so patterns of sequences, again, to kind of highlight the point, all of these different types of sequences, as you can see, if black and white dots represent hydrophilic or hydrophobic, you can see that the patterns for globular membrane and fibrous proteins is quite different, but it always will lead to about 50-50, which is good because we just said that we have about 50% of all the amino acids are hydrophobic or hydrophilic. So whereas globular proteins are mostly kind of a random assembly of these proteins, in case of membrane proteins, it's really not the case. We need to have all the hydrophobic residues that are sitting together because then they will form helices that will be in the membrane, remember? While hydrophilic residues will be mostly in the loops, and so that's why they are together in terms of primary sequence. In one of Eric's lectures, you talked about fibrous proteins. Remember, that was, I think you talked maybe about silk and collagen, and also maybe the proteins that cause neurodegenerative diseases. But anyways, those are kind of remarkable because they're made of repeats, so identical patterns of residues, quite different from the globular case. And this is because their role is only to structure, so it will be small units that are just repeated on top of one another, and you remember they were creating kind of in collagen, the basis for example, hair structure and so on. Or in silk is the same. So we've talked about this before. It seems really that there are very few defects. It seems that in proteins we can lose maybe 1 or 2 hydrogen bonds, but often not much more than that. And the question that we're going to be answering over the next 20 minutes or so is, why is that? Because do you remember this number? Losing 1 hydrogen bond costs about 5 kilocalories per mole. It looks quite small in the big scheme of things. So why is it so rare that we see them? So 2 hydrogen bonds would cost 10 kcal per mole. This looks like a small fraction of the total energy if you consider all of the hydrogen bonds that are formed in your protein, right? So it doesn't seem like such a big deal. We're going to come to that. Similar idea, you've seen this picture before. Maybe not this one, but you talked about this. Beta sheets can really only be right-handed crossing, not left-handed, so it's a similar story there. So why do we not afford defects? And we're going to talk about the balance between enthalpy and entropy. And I already talked about this in the simple physics model. The folds that can choose between many conformations can accommodate more sequences in low energy states. So if you have a protein that can assemble in slightly different ways, then it will be able to get different conformations, and then you could have many different sequences in there. So we know how to talk about energy, I think, or you guys know, and that is related to the Boltzmann statistics. I think you derived this formula. Basically what we said is that the occurrence of elements in proteins, this probability density, is proportional to the exponential of minus delta E over KT, where delta E would be difference in energy, and KT would be setting the scale for energy in physics, K being the Boltzmann constant and T the temperature, right? And we see that this holds experimentally whenever you see 2 things that are separated by more than KT, then there's a relationship that looks like this, and it means that the thing that is higher in energy by more than KT will never be observed, or will be observed with a probability that is very low because of this exponential difference. But there's a caveat here. And it's that we can, in fact, not use this relationship, and we're going to derive which one we can. The reason we can't use this relationship here is that this all relies on the fact that your protein can sample a variety of states. So you might be comparing a folded and unfolded state. Then you can apply Boltzmann statistics. But the changes in sequences, they don't interconverge, so you can't explore all of the possibilities of sequences because it's a driven process by evolution. So this means we can't use the Boltzmann distribution. The structure is constant, and we will ask why many sequences can fit it. So this is all summarized in the multitude principle, which you might want to memorize. The more sequences can fit a given architecture without disturbing its stability, the higher the occurrence of this architecture in native proteins, which in other words means that defective patterns are not impossible, they're just quite rare. So I'm going to give you 1 minute to go through this, and if there's still a problem after this 1 minute or 30 seconds, then ask me a question. We're going to use this in what comes. Yes?
What do you define by a defect?
By defect, I mean these unpaired hydrogen bonds. So again, remember my argument that you couldn't have one beta strand surrounded by helices because it would leave exposed many carbonyl and NH groups. An exposed carbonyl group that is not paired, that's a defect. Or you could have a single polar group that is in a hydrophobic environment, or a single charged group. All of those are defects, so irregularities that cost energy. Any other questions on this?
How do you derive that from the thing in white, the text in white?
How do we derive the red?
Yeah.
This is just a comment. It's about the higher the occurrence, so it's not all or none. It's not that you will never see defects. So see, this is phrased probabilistically. The more sequences that can fit a given architecture, the higher the occurrence. It's only a little comment just so that you guys don't remember. It's unheard of to have a hydrophilic residue alone in a hydrophobic environment. Okay? So here's our little model where the equations come. You might say, finally, for those of you who want to see equations. It's not there, it's on the next slide. Why do we have a limited number of folds for globular proteins? We have approximately equal fractions of hydrophobic and hydrophilic residues. And how well do such sequences fit the folds and secondary structures we see? So reminder, your alpha helix, we'll talk about the alpha helix specifically. We won't talk about the pi helix, which would be this one, but the argument would be similar. And in this kind of structure, you have a hydrogen bond between amino acid I and amino acid I plus 4. Remember? And each turn of a helix takes about 3.4, 3.5 amino acid. In a beta sheet, you have an alternation of two amino acids from one plane to the other. And the repeat is made of two amino acids, right? One up, one down, and then you go to one up, one down. So the pattern here is slightly different. For one structural turn here, one structural element is 3.4 amino acids, and on here it's two amino acid. So the question we're going to be asking here is, what's the typical length of a helix, and when does the helix or the sheet break? Meaning, when does it go into a turn? So if we call P the fraction of a specific thing in nature, let's reason on the polar versus nonpolar for the moment. So if we call P the fraction of nonpolar residue in the sequence, what is the average number of such groups we will find in a given stretch? Well, let's say these are all my hydrophobic residues in red and my hydrophilic residues in blue. So nonpolar is synonymous with hydrophobic. then the probability to find these groups in a stretch will be the probability for this blue one is 1 minus p. Then the probability for this is p, 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 so p to the r times, right? And then finally it's broken by another amino acid with probability 1 minus p. So then these are all independent from one another. So to get the probability of the whole thing, we just multiply all of these probabilities together, and we find that the probability W of having R residues of character P in a row is this. So remember I was saying what we're interested in is what's the typical length of such a structural element, right? So to calculate this, we need to calculate the weighted average of this. So R was this length. And this will be given by this formula. Do you recognize the formula for the weighted average? You take the value of the value of the quantity you're trying to calculate the weighted average of R. You multiply it by the probability of that specific value, and you sum over that, and you divide by the sum of all probabilities. Yes? And the reasons why these sums start at 2 and not at 1 is that having just one structural element is not interesting. That would just be alternation. So we start at 2, meaning having 2 in a row that then breaks. And if in the book it says go to your class on what they call on series, and you'll find it doesn't even tell you where to look it, but apparently it's a known relationship. If you take the sum of p to the r, then you can write it summed over r from 1 to n, then it goes to this number. And so if you then solve for this using this relationship, you'll end up with the notion that the average length of such a I can't talk anymore of such an element is 2 plus p over 1 minus p. By the way, I guess you guys have seen this. I should have said from the first element, these 1 minus p's up there, they disappeared because they're both the same at the top and the bottom, right? And they don't depend on r, so that's why they vanished. It's a little bit more detailed in the book in case you need to go through the derivation. But what I'm interested in now, if you trust this whole expression, we'll end up with r is equal to 2 plus p over 1 minus p. And so if we have a probability that is 0.5, then the typical value of an arrangement that is of length will be a length of 3, right? 2 plus 0.5 over 0.5 is 3. But is that what we see in nature? Do we see 3 amino acids in a row and then a break? Usually not, in fact. Usually what we see is that an alpha helix is often 11 residues long, and 11 is 3 times 3.6. And remember, I was telling you 3.6 is the helical turn. And beta sheets are often 6 residues long. And 6 residues long is 3 times 2, and 2 was the typical unit that we found in the beta sheet, right? So it turns out that if we use this simple model and we observe nature, then we find that this p that I've been using so far should be the probability of 3.6 amino acids to be in a helix, or the probability of 2 amino acids to be in a beta sheet instead of the hydrophilic versus hydrophobic propensity. So that actually puts constraints on secondary element size. It looks like the size of these things are governed by physics. Quite simply. Exactly. You can, according to the book, do a similar derivation. It doesn't list how, so I assume it's not required for you to understand how. you can derive the average loop length this way, and this will come out to a slightly different formula, 3 plus 1 over 2p squared. And this you will find now that with using p equals 0.5, you'll end up with an average loop length that is 5. And 5 is a typical loop length. So if you take all of this together, it looks like from this very simple model, you can derive the typical fold. So remember what I told you, right? You had either helices or beta strands and loops taken together. This model derives how long they can be. And then physics, whether they're hydrophobic or hydrophilic, will determine how they can arrange with respect to one another and to form 2 or 3 layers and not less and not more. Yes? So with all of this taken together, now we're really starting to have a lot of constraints on what the folds can be, I hope you can see. And that means that this, when we went through these 3 possibilities, we had the physics-based thing with the question mark in the middle. So now we're really putting constraints based on physics on what can happen. Okay, so back to our stabilities now, and why the Boltzmann distribution will not work out. There's an interesting relationship between the transfer free energy from alcohol to water and the occurrence in proteins. This is on a log scale, how much residue is found in the core and on the surface. And there's a correlation, and of course it makes sense because for globular proteins we want to embed hydrophobic residues and have hydrophilic residues around them. And you can see these are all the amino acid single letter codes. And you can see that there's a almost linear relationship on this log scale, right? So if that's a log scale, this means that there's an exponential relationship, again, between occurrence and transfer free energy. So this is not directly related to the point of defect. It's to show you that, again, there's an exponential relationship in coming up in biology. And this will be also true for rotamers, occurrence of ion pairs, and so on. So what is the relationship if it's not the Boltzmann relationship? What is the exponential relationship that we will have when it comes to possible folds and accommodating defects? Here's the reasoning. The native state is stable if the free energy is lower, of the folded state is lower than all the other states, right? We need to be in this. We need this to happen for the protein to fold so that it can then perform its function. From the previous slide here, I saw that serine that the transfer from oil to water of a serine will be 0 kcal per mole, whereas exposing a leucine from the inside to the water will incur a cost of 2 kcals per mole. So again, these are small numbers. We talked about hydrogen bonds, 5. Now this is another type of defect, 2 kcals per mole. It looks small. But what this means is that we will be able to fold a protein with a leucine that is exposed to the solvent, but it will cost something, so there will be fewer folds. I don't understand this anymore. There will be fewer folds that enable leucine to be outside. The total free energy of the fold, if we have this defect, will be the energy of the free energy of the rest of the chain plus the energy of that defect.
So this is wrong when it said but fold with leucine works for non-sequences, or it's right?
But fold with leucine. I think this argument was, I'm going to read my notes. The leucine is hydrophobic, so prefers to be inside. So it's possible to fold a protein with the serine inside, but the leucine will always win in this position. So there will be more folds with leucine inside. I was making the opposite argument. I was talking about putting the leucine outside. So it's just the reverse argument. You win by burying the leucine, you lose by by exposing the leucine. So it always depends on the sign here. So I think these slides don't make sense because this is a positive number, and now it's telling you what happens when leucine is buried. So I think it would make more sense if we put a minus side here or if we use the reverse argument. So what I said first was correct. Thanks for asking. So what you need to focus on for the next slide is that the total energy is delta F plus delta E for this protein in which we have put a defect. And this is an empirical derivation. If you look at the ensemble of energies that are possible for a large number of sequences, they will assume a Gaussian distribution. And why is that? That's simply central limit theorem, the law of large numbers, right? If you have many possibilities, so you take your whole sequence, and at each position you can have the 20 amino acids. It gives you a large number of possibilities, and you calculate the free energy of this specific fold with all these possible sequences. Then the distribution will look like this. What is really quite essential here, and so I should say Gaussian distribution with mean delta F and standard deviation sigma squared. What is quite interesting and kind of maybe unexpected is that the center of this distribution is well above 0. So see, this means that all of these sequences are unfavorable. So there's only a very small number actually of sequences that are in the allowed regime and below 0. So the argument will come as to why so few defects are possible. We said before that the total energy will be delta F plus delta E. So to accommodate a specific defect that is delta E, I can only have all the sequences that are here in this black part, because adding delta E will bring me to 0. So if my energy before inserting the defect is right here, adding delta E will push me above 0, and then I will not observe this sequence, right? So the number of allowed sequence that would enable to accommodate the defect is really this black side. And this, to calculate then the probability that delta F is lower than delta E, or in other words, the probability that delta F plus delta E is lower than 0, is then integrating from minus infinity to minus delta E, this probability density function, which is a Gaussian. And this is easy to do because it's a Gaussian, so integrating a Gaussian is quite simple. And in fact, the fold, this will come out with the fact that the fold is stable if delta F is lower than minus delta E, and the probability of this is equal to a constant times exponential minus delta E over this factor that is sigma squared divided by delta F, so variance over the mean. So do you recognize features in this? Remember the Boltzmann distribution was C e to the minus delta E over kt. So there is something similar in the form, but the normalization factor here is not kt. It's this sigma square over delta F. And that's why we will be able to see these exponential kind of relationships, but that we don't follow the Boltzmann distribution. And again, this is related to the argument I made before the break, that, or maybe after, I don't remember anymore, that the Boltzmann distribution cannot be used because we're not sampling exclusively all the sequence space because we're guided by evolution. But basically from this, it means that if I increase delta E, I'm reducing the number of stabilizing sequences exponentially. Okay? So again, inserting a small defect will make the number of possible sequences that I can use very, very small. So let's talk about this a little bit. What does this normalization factor, sigma square over kt, do? This we can, did I write this somewhere? Yes. This serves kind of as a reference, and this could be called as the characteristic energy, because we need to compare delta E to this number to say whether it's allowed or not, right, in an exponential form. You may recall that sigma square and the mean are proportional to the size of the sample, but the ratio of sigma squared over delta F is size independent. So this means that this characteristic energy will not depend on the total size of the sequence. And this yields a really interesting argument, because whether the protein is 50 amino acid or 500 or 5,000, we have the same stabilization energy. But it's much more probable to insert a defect in a long sequence, right? And this comes back to the notion that it's much easier that the protein arranges in small domains of 50 amino acids, because you have much less of a probability to insert a defect into a small domain as opposed to a big one. That's why nature tends to have small building blocks, because big domains are difficult to fold without making a mistake and inserting a defect, and that would then push your energy above this 0 format. Yes, this could be, I'm coming, it could be a lot to take in, but the argument is there's really not much in there. If you look at just read the page in the book, and you'll see it's quite simple. It's really about understanding this Gaussian distribution. Yes?
No, I found it.
Okay. Do you want to still say it? Because maybe other people were confused. This KTC? Yeah, so exactly. So in the Boltzmann distribution is KTC, and we call it the characteristic energy, and here instead is sigma square over delta F. So conclusion of this part is that most sequences will not fold into stable structures. Only a subset will, and only an even smaller subset will accept mistakes. And we just derive this quantitatively. I think you've seen this maybe before. It's another small thermodynamic argument. Here's a quiz to wake you up. Which is more common? We've seen this before, so you need to be able to answer this, because we've seen this in this lecture. Which is more common in the protein inside? Sheets or helices? So is it more common to have sheets with helices on both sides, or is it more common to have helices with sheets on both sides? Sheets with helices, raise your hand. Helices with sheets? Yes. So remember the Rossmann fold, right? We had the helix and the sheet and the helices in the way. We can make an argument here that is entropic in nature. Helices, one helical turn takes 3 to 4 amino acid, whereas the beta strand only takes 2 amino acids. So there's more ways to make, to arrange smaller things in the middle. And that's why there are many more, that's why it's more favorable to put beta strands in between alpha helices. Just a little story about biology. to relax you. There is one exception to this, and in a way it's unfortunate because it's very prevalent. Just to make you think, remember I was explaining to you that we can't have one beta strand in helices. How about this one? Is that as unfavorable, having one helix in a beta barrel? Do we have exposed hydrogen bonds in an alpha helix? We don't, right? Everything is paired. So this does not make any defects on the side. So that is actually thermodynamically possible. It's unusual, but it's not as forbidden as the previous case. So this is GFP. It has a little cute name because it's so much used, green fluorescent protein. And basically, this is the shape that it has. So it's a beta barrel with a helix in the middle. And it has inside a so-called chromophore, which is a chemical that is able to absorb and release light. The typical protein absorbs blue and ultraviolet light and emits green light. That's why it's called green fluorescent protein. And researchers have designed synthetic, so this occurs in nature actually in jellyfish. But researchers have designed chromophores that are slightly different and that can absorb and emit light that are in different kind of color regimes. What's going on? Ah. So like I said, it's found in nature. I don't know what this is. It was described by this guy that Roger Tsien that died quite young, actually, kind of tragically. And like I said, they were able to make a GFP of different colors. So these are all bacteria that express these different types of GSP. So you can just make nice patterns like this. And either that having fun in the lab, it's quite used to treat for surgeons. So this is a view of what a surgeon sees when they're trying to remove a tumor in ovarian tissue. And if you look under white light, I mean, surgeons are trained, right? But it's really quite difficult to know what you want to remove or what you don't want to remove. And now what we can do is that we can label the cells of the tumor in a specific color. And then you can label the cells of the tissues that you don't want to touch in a different color. And by superimposing these two things, you can really just extract the tumor thing. So very practical application for this unusual fold of a protein. So these would be, I think, nerves, and this is the tumor. And see, you can, if you're a good surgeon, I assume you can really kind of go and not touch the nerve, only the tumor. Yes? I think by that point, the cancer is worse. For the nerve. So I'm not sure. So my cell biology is not great, but proteins like routinely get degraded, right? This protein doesn't have another function than emitting light, so it's not toxic per se. I guess it might be toxic over long times, but in fact, it will get degraded over time and then replaced by, natural GFP. So I'm also not sure. I think this is done with viral injection, so this phage kind of thing where, they use the viruses to inject the cells. But I think this has been approved by the competent authorities who have determined that it's better to do this than to not do it. Yes? It's with this fluorophore. Again, not my specialty. Apparently, I'm not a specialist of many things. These are conjugated pi systems, right? With are you guys chemists, any of you? No? So I can say anything. So there's two pi clouds, and it's this thing with the Jablonski diagram. I mean, it's a whole class on kind of fluorescence, but basically when there's two conjugated pi systems, inserting lights just changes excites the electrons to another level, and then it relaxes to a different kind of state that is slightly lower in energy, and then the photon gets expulsed as it returns back to the ground state. And so that's why the light color is slightly different from the absorption to the emission. And by changing the chemistry, you can change basically the color that will be absorbed and emitted, because the energy levels of the electrons on the two molecules are slightly different. It's pretty cool. I suggest studying fluorescence if you have time, because then you can learn more than what I can tell you. Okay? So we have 10 minutes. I will definitely not reach the end of this, but I'm still going to go on for 10 minutes to give you as much information as we can. And this whole part is attempting to derive arguments about folding and whether so we've determined that the units are small folds, right? And that we will have not big proteins with many layers, but instead we will have small domains that kind of come together. And this whole section is about deriving whether these domains fold into one cooperatively very quickly, or whether there's a gradual way that it finds itself. But before we talk about this, we need to make a quick detour through molecular interactions, because in order to explain the type of curves that we'll see, I need this. I think maybe Burke went through this briefly, but if not, it's an interesting kind of topic. So recognition of ligands by proteins is, of course, very important for biology in general. And there have been, over the years, 3 main different models that were developed. This one is called lock and key. So this was the idea that there would be a very specific pocket that would really fit the specific ligand shape, and that different proteins and different ligands would be very tailored for one another. Of course, this is a bit idealistic. Biology never looks like that, but I guess when they were making these models, we didn't know so much. This is called induced fit. It's the opposite idea. The idea is that the protein takes in whatever conformation that is not so compatible with ligand binding, but when the ligand comes, then the protein binding site forms around the ligand. So it's called fit, and it's induced by the ligand binding. And then the selected fit kind of model is a mix between induced fit, which is what I just described right now, and conformational selection, which involves the protein moving in between conformational states, and the ligand only binding when the state is visited. So there's one that is not compatible with ligand binding, there's one that is, and then through induced fit, the pocket forms around the ligand. Through conformational selection, the ligand selects for the conformation only that was compatible with ligand binding. It turns out that reality is really a mix of these two pathways. And I mean, these schemes are nice, but I guess you guys by now have really seen what these domains look like and how complicated they are and how dynamics comes in, so you shouldn't be surprised that this happens. So this is an example of selected fit. We have the red protein that is unbound and the blue protein that is bound. I'm sorry, I should say state. Around this yellow ligand, you can see that the blue pocket is slightly more closed than the red one. And this is a 6 angstrom motion. Remember from the labs, is that big, is that small? It's kind of big, right? We saw that okay, it's not huge. I see you're disappointed. It's not huge, but you can definitely see that there are several structural elements that are moving with respect to one another. So in order to accommodate the ligand, this takes a bit. Reminder to remember what the RMSD means. So we're back to our hemoglobin, which was, remember, it was 4 times a globin. It binds 4 hemes that will bind oxygen in each of the domains. And this is the curve that shows how saturated this protein is with oxygen pressure. And the red curve is for hemoglobin. So this kind of curve shows you that as the oxygen pressure increases, you don't bind a lot at all initially. It's kind of a weak binder. But as the protein binds the first oxygen, it undergoes a conformational change in the other domains, which make it much better at binding oxygen. And the third one is even faster until you reach saturation. And this kind of curve is really this kind of sigmoid shape, S shape, really is kind of crucial in biology and I guess to some extent physics and chemistry, and displays cooperativity. See how myoglobin up there is completely different? Myoglobin is a good binder, but binding one molecule only reduces how much you can bind of a second one. Yes? So this illustrates the conformational change that is going on when you're in this kind of regime. It's also called allosteric modulation. Allosteric modulation means I bind in one domain and I cause a conformational change at a distance. Allosteric means at a distance. And as this allosteric change is going on, then the conformational change changes the affinity for the oxygen molecule. So I really went through this to show you a cool example, but also to highlight this cooperativity mechanism, because now we go back to our question of folding and unfolding. And we're looking here at something that measures folding, you don't have to know what, as a function of a concentration of denaturant. And you can see that as you're increasing the concentration of denaturant, you are unfolding, but you're not unfolding gradually. Recognize this S shape, right? When here you don't do much, and then as you start unfolding, then it goes very fast towards very rapid unfolding until you reach the completely unfolded state. So this tends to say that our process of unfolding is going to be cooperative. And again, I'm not talking here about kinetics because you've seen this with Burke. So this is really about thermodynamics and measuring the steady state properties at the end of the transition. So this was in the presence of a denaturant, but other physical things that cause denaturation or unfolding, like pH temperature, they cause the same type of transition, which looks like it's cooperative. This guy, Anfinsen, was the first to show that